We treat DNA as a rigid architectural blueprint, immutable instructions mapping our lives with mathematical precision. But our organic reality is much more complex. This chart breaks down the architecture of a 1 billion word text. These 4 nucleotides, A, C, G, and T, sequence the entire record of our biological history, letter by letter. This fundamental alphabet is applied so universally across nature that 98% of our sequence is identical to that of a chimpanzee. This staggering overlap raises a question about our autonomy. If our letters are nearly identical to a chimpanzee's, we need to determine exactly how much control those letters exert over our behavior and our future. In some parts of our genome, the control is absolute. Biologists call this hard determinism. Specific regions on chromosome 4 where the genetic code leaves no room for environmental influence or personal choice. This diagram illustrates the onset of Huntington's disease. It is triggered by a microscopic stutter of CAG repeats that acts as a rigid, inescapable script within the DNA. This 1-inch section of code determines a fatal outcome with absolute certainty. No lifestyle change or environmental factor can override it. If our bodies carry such unyielding commands, it's easy to wonder if we are simply passive biological machines, playing out a destiny that was written long before we were born. The spectrum of genetic fate replaces the idea of total control with a gradient. Chromosome 6's IGF2R gene provides a baseline potential our environment must actively engage. Further down, chromosome 17's TP53 gene acts as a defense system. When it fails, it creates a probabilistic risk for cancer, rather than a guaranteed sentence. This web of interactions on chromosome 1 dismantles the myth that a single gene controls a single trait. Physical outcomes, like the health of these lungs, depend on a pluralistic network of multiple blue genetic nodes and green environmental factors working together. Biological traits are not solo performances dictated by a single gene. They are complex symphonies that require both the genetic instrument and the environmental player. This interaction is a two-way street. Our psychological urges and conscious behaviors can force direct physical reactions within our biology. As shown in this spark panel, the D4DR gene on chromosome 11 influences dopamine processing, creating the psychological drive behind thrill-seeking personalities. When acted upon, it triggers a cascade on chromosome 10, resulting in the physical hormonal response mapped on the right. Over thousands of years, these willed cultural choices have permanently altered our species' genetic composition. This chart tracks how the willed action of consuming milk led to a permanent dietary shift. Over generations, this cultural choice drove the biological evolution of lactase persistence. Our biological reality is suspended in the gap between these two forces. We have a determined ancestral foundation on one side, but our willed expression ultimately governs how that text is performed. Humanity has reached a threshold. We have moved from observing our genetic sequence to surgically intervening within it. This represents the era of precision editing. Using CRISPR-Cas9, we can execute find and replace changes directly on the human manuscript, treating DNA like a digital document. We are also exporting the very architecture of our biology into our technology, creating a bridge between the human genome and artificial intelligence. This parallel shows how researchers are mapping discrete computational behaviors, like reasoning and memory, into AI chromosomes to sequence machine behavior. This timeline shows our transition from carrying the text to rewriting it. By merging our biological code with silicon architecture, we have taken direct control over the future trajectory of our evolution.